Hello, guys. So today I'm going to be starting IB biology. As you know, IB biology is a bit of a dense program. We really delve into the details, but if you actually watch this video, then you're actually going to be knowing every single detail of section 1.1. And at the end, we're going to be practicing paper 2 questions. So by the end of this video, you're going to be very, very, very understanding of every single detail of this section. So let's begin. So firstly, what is a cell? The cell is the basic unit of life. It's the smallest part of the organism which is living, and it carries out essential processes of life. Now, we do know that essential processes of life mean metabolism. So once they ask you which function of life indicates that the organism is alive, you always say metabolism, because metabolism is when the living thing undertakes essential chemical reactions. So usually this is asked in the paper 1 questions. If you don't know what paper 1 questions are, they are MCQ questions. So now let's begin. The principles of the cell theory. This is very, very important, and you should actually know this by heart because it's asked about a lot in the IB. It's one of the top asked questions. So let's begin. So the first tenet is all living things are composed of cells. Cells are the building block of structure in living things, meaning that living things are made out of cells. We are composed of cells. The second tenet is the cell is the smallest unit of life. The third is cells only arise from pre-existing cells by division. Now, why did I write mitosis? Because sometimes in these paper 1 questions, what they do is that they give you like a number of statements, and they're like, which statement or statements pertain to the cell theory, and sometimes they say mitosis, but it's not correct. Sometimes it's not always mitosis. So you have to be careful. If it says that cells only rise from pre-existing cells by mitosis, this is false. Do not put this as a correct answer. Now, 2 new concepts to the theory. So these are new. They are not completely part of the cell theory, but they are new. So cells contain a blueprint of information for the growth, development, and behavior, and cells are the site of all chemical reactions of life, metabolism. So usually when they ask you to say the cell theory, you actually just say these 3. But if they ask you about the 2 new ones, then that's when you say these ones. Now let's go to the functions of life. And so we have the functions of life. Now you have to know how to state them, but you also have to know how to outline them, meaning that you have to know what they are, but you actually have to know how to define them as well. So what the trick I use is I use the Mrs. Hang trick, which is basically I just remember Mrs. Hang, and that's how I remember all the functions of life. So firstly, we have metabolism. It's when the living things undertake essential chemical reactions of life. Reproduction is when living things produce offspring sexually or asexually. Now, what the difference between sexually and asexually is that it's in sexual reproduction, fertilization takes place where there is a male gamete and whereas there's a female gamete. Whereas in asexual reproduction, there is no fertilization whatsoever. In sensitivity, living things respond to internal or external stimuli. In homeostasis, living things maintain stable internal environment. In excretion, living things exhibit the removal of waste products, such as feces. Nutrition, living things exchange gases within the environment, and with growth, living things can move and change in shape or size. So remember, you have to state them and you have to be able to outline them as well. Now we're going to be talking about both the paramecium and the chlorella. Why are these important? Because we usually talk about these two in order to compare. So firstly, we have the paramecium, and I'm going to be following the Mrs. Hang. That way, it's really, really much easier to remember, at least for me. So for the metabolism, most metabolic processes, metabolic pathways take place in the cytoplasm. So as you can see, here is the cytoplasm. It obtains biochemicals that requires through the digestion of food particles. As you can see, these are the food particles. And now we're going to include movement, even though movement is not necessarily a function of life, but we will discuss it as well because it's discussed in many IB resources, so it's best to include it. Basically, it swims rapidly through water, rotating. Food vacuoles can be seen carried around the cytoplasm. And now, we're going to be discussing reproduction. The nucleus divides to support cell division, followed by transverse of cytoplasm. And keep in mind the reproduction is asexual. For the response, the wave of action of the cilia. Now, this is the cilia. They're like hair-like structures, and they help in the movement of the paramecium. So basically what happens is that the wave of action on the cilia move the paramecium in response to prey. Maybe it sees a predator, or maybe it just needs to leave a certain environment, and the cilia helps it do that. And after that, we have the respiration. So basically it respires aerobically, thus transferring energy to the cell. Now you have to remember, I'm going to be very brief with this because this is not something you should know for this chapter, but basically respiring aerobically means that it requires oxygen. And also a product of aerobic respiration is ATP, which is energy. That's why it transfers energy to the cell. So just keep that in mind. And now let's move on to sensitivity. So it typically detects favorable food particles and moves towards them. These are the food particles, as you can see. Homeostasis, contractile vacuole. This is the contractile vacuole. Fills up with water and expels through the plasma membrane to change the water content. So basically what the plasma membrane does, that it controls what enters and what exits the cell. Excretion, the plasma membrane controls the entry and exits of substances, including the expulsion of metabolic wastes. Nutrition, it's basically the paramecium itself is a particle feeder. It takes in small floating unicellular organisms. So basically unicellular organisms are organisms that are made out of one cell, whereas multicellular organisms are organisms that are made out of more than one cell. Keep in mind that unicellular organisms are able to actually maintain all the functions of life, even though it's just one cell. Into food vacuoles in the cytoplasm, where it controls and digests where contents are digested and products are absorbed. So this is basically here. It's when the nutrients are absorbed into the cytoplasm and there's enzymes that digest the food particles. You can see here. So growth, small cells go to full size prior to cell division. And again, you have to always know the definition of a paramecium. It's a large protozoan common in freshwater ponds. So now let's go to the chlorella. So firstly, what is a chlorella? A chlorella is a unicellular green algae. So first, we're going to start comparing here. So firstly, when it comes to metabolism, most metabolic pathways happen in the cytoplasm. So this is something they have in common, but it manufactures its sugars from photosynthesis. So basically, it has a chloroplast, which contains the green pigment chlorophyll, and it's able to actually undergo the process of photosynthesis. That means that they do actually have a different type of metabolic processes. So we have to keep that in mind. And don't focus on photosynthesis. Again, this is in chapter 2.9. So we're not going to focus on that much. And ions from nitrate ions surrounding it in water, and energy is transferred from respiration, making it possible again. And now for movement, the cytoplasm within the stream around within the cytoplasm. Then we have reproduction. The cell contents divide into 4 autospores. So this is it. And each form a cell wall around them. And then they're eventually released by the mother cell wall. So this is it. So this is like how it was one, and it divides into 4 autospores. So basically the reproduction is also different. Now for respiration, it respires aerobically, again using oxygen, and it transfers the energy to maintain cell function. So they have the same respiration, even though if they tell you what common function of life do they have in common, do not say respiration, because it is not a function of life. And then sensitivity, it responds to the absence of light by nuclear. I'm sorry, I didn't write nuclear division, but it is nuclear division followed by cell division. So first the nucleus divides and then the cell divides after that. And then homeostasis, the contractile vacuole fills up with water and expels through the plasma membrane in order to manage the water content. So they actually do have homeostasis in common. The excretion, so loss of waste products, mainly CO2. So basically you have to know about photosynthesis, that that CO2 is actually a waste product of photosynthesis, but don't actually focus on that for now, because this is for chapter 2.9, very, very later on in the program, through the entire surface of the cell. Nutrition, photosynthesis takes place inside the chloroplast. Like I said, you have to have a chloroplast for photosynthesis to take place. Manufacturing sugar from water and carbon dioxide, because water and carbon dioxide are actually raw material of photosynthesis. Make sure you know that. To provide the algae with food almost identical to flowering plants. And then growth, cells grow into full size prior to division into autospores. So again, their growth is also different. So keep in mind that their homeostasis is the same, but everything else is quite different. So now let us discuss the surface area to volume ratio. So firstly, we have to know that the plasma membrane is responsible for whatever goes in and outside the cell, export and import basically. And all of the metabolic reactions take place inside the membranes. What the mass or volume is, it's basically the rate of the metabolism of the cell. So basically a larger cell needs more energy to sustain essential life functions, whereas the smaller cells, they need less energy, making them have a higher surface area to volume ratio, because usually these smaller cells, they're more efficient and they're faster. Surface area, the rate of surface area is the rate of material exchange. So basically larger membrane surfaces equate to more material movement. So they need more material movement as well. So more time and more material movement. So as you can see, there's less and less efficiency the larger the cell is. Therefore, growing cells tend to divide and remain small in order to maintain a high surface area to volume ratio, because they need to survive. And now these are the formulas. So basically total surface area is when you multiply the height by the width, by the side, by the number of boxes, whereas the total volume is height times width, but instead of sides, you have to multiply by length times the number of boxes. Now, the surface area to volume ratio is basically you just divide the surface area to the volume. It's a ratio, you just divide them. And you have to keep in mind that a large surface area to volume ratio means that the cell can act more efficiently. For every unit of volume that requires nutrients or waste or product or produces waste, there's more membrane to serve it. So basically, the higher the surface area to volume ratio is, the more there's actually the more efficient it actually is, basically. And the volume increases faster than the surface area of the cell, and less cytoplasm has access to the cell, causing exchange of gases, supply of nutrients, and losses of waste product. And the smaller the cell is, the easier it is for the cytoplasm to exchange material and gases with the environment. As you can see, again, my point, efficiency. When the cell reaches a maximum volume, it actually divides in order to, survive. So this image is actually taken from Bionology. So firstly, this just shows that in a smaller cell, there's a shorter diffusion distance, meaning it takes less time. So when diffusion pathways are shorter, molecules don't have to travel to get in and out of the cell, basically making it take less time. So, but there's a downside at sometimes, like there may be some disadvantages to having a large surface area to volume ratio, because for some specific species or organisms, it's not an advantage. For example, for small warm-blooded animals, what happens at these animals, these creatures, they lose heat quickly. So what happens is that because of their large surface area to volume ratio, they end up having to eat constantly, which actually may be a disadvantage because they're constantly losing heat. And desert plants would lose their water quickly, so they have to minimize their surface area to volume ratio, because they actually need to conserve these water, because they actually need the water for survival. Now, as organisms grow, cells divide. So what happens when these cells divide? Firstly, there's cellular differentiation. These cells, they actually start becoming specialized for certain and specific functions, like I said, specialized functions. And there's a complex multicellular life. So basically, we're going to be talking more about that when we talk about emergent properties. Now, cells compartmentalize, meaning they use the membrane to carry out metabolic processes. So in eukaryotes, we're going to be talking highly or like in detail about eukaryotes and prokaryotes in the next video, 1.1, 1.2. So don't focus on that for now. But organelles are made out of membranes, maximizing the surface area of reactions, and some organs fold up to maximize their surface area to volume ratio, and that actually can be seen with intestines. And actually, if you Google the intestines, you can actually see the shape itself. Now, we're going to be comparing animals and plant cells. So first, we're going to be talking about what they have in common. So firstly, they both have nucleus, they both have cytoplasm, and they both have plasma membranes. But animals have centrosomes, and they have temporary vacuoles. But plants don't have centrosomes, and they have large permanent vacuoles. Furthermore, animals have secretory granules, and they store their food as glycogen. Whereas plants have cell walls that are made out of cellulose. Us humans, we cannot digest cellulose because we don't have the enzymes to digest the cellulose. Plants have chloroplasts, but specifically the ones that undergo photosynthesis. And they have starch, and they have large permanent vacuoles, like I said, and they're spits where the cytoplasm and the cell connect. So as you can see, these two cells are quite different. Now, do not delve deeply into these because these are not usually asked about in the IB questions, but of course, you never know what's going to come up. And you also should always know these in order to understand and build on your knowledge. Now, the origins of the cell theory. So firstly, we have Zacharias Jensen, where it all started, or at least it first became recorded. So in the year 1950, he invented the first compound microscope. In 1965, Robert Hooke devised compound microscope and used it to actually observe the structure of cork. He drew cork cells and then he measured them. And then he ended up coining the word cell, and he was the first to use it. In 1673, Antoine van Leeuwenhoek, sorry if I didn't pronounce this correct, but he developed a hobby of making lenses, and then he mounted them in metal plates to form a simple microscope. Magnifications of 240 were discovered. We're going to discuss magnifications later. And he was the first to see living microscope organisms such as the sperm cells, protozoa, and bacteria. Robert Brown, now it's not known which year he made his discoveries, because it was not in the source I found, because again, these information, they're from very, very, very different sources. So some things, and they're really, really like mixed up because they're from plenty different sources for you, in order for you to be able to get as much answers correct in the mark scheme. Now, Robert Brown, he observed and named the nucleus cell, the nucleus, and he observed random particles such as falling grains when suspended in water. Now we have Matthias Skildian and Theodor Schwann. So in 1838, Matthias concluded that all plants are made out of cells, whereas Theodor, he concluded that all animals are made out of cells. Now, both of them, that's why they're always together, because they're both credited for developing the first two tenets of the cell theory that we just discussed previously in the video. And they both established that cells are organisms, and entire animals and plants are aggregates to these organisms arranged to the finite laws. And then Rudolf Virchow in the year 1855 established the idea that where a cell exists, there must have been a pre-existing cell. So therefore, he was credited for developing the third tenet of the cell theory. So the first two were Skildian and Schwann, and the third one was Virchow. And then the last one was Louis Pasteur. He established the idea that life does not spontaneously generate. But this you don't have to focus on, at least for chapter 1.1, section 1.1. We'll discuss this thoroughly in section 1.5. So now microscopes. So a microscope, firstly, what is it? A microscope is a scientific instrument that's used to visualize objects that are too small to be seen with the naked eye. Now, there are two types of microscope that we discuss in IB. Firstly, we have the light microscope, and second, we have the electron microscope. So in the light microscope, we're able to see living specimens in natural color, whereas in electron microscopes, electromagnets are used to focus electrons, resulting in signifying greater magnifications resolutions. So basically, what is meant is that you're able to see things more clearly with a better view. And this is the reason why the electron microscope can actually be used to view dead specimens in monochrome, meaning that sometimes the color may be like false. The color that's applied may be false. They're not accurate, the colors. But what resolution means, very important to know what resolution means, because it actually really asked about in the IB questions. It's basically the ability to separate two objects from one another. So basically, if two objects cannot be separated, then they're actually considered as one object. And if the limited resolution in the light microscope is 0.2 micrometers, and the chemical dyes and fluorescent labeling may be applied to resolve specific structures. So as you can see, this is the difference between the two microscopes. But you should know that the electron microscope can actually see dead specimens, whereas the light microscope can only see alive specimens. Now, let's discuss magnification. So now I'm just going to give you this really, really good trick I learned. Always, oh, sorry, here. Oh, I actually did it correct. Never mind. Always do this. Here, write I, A, Why? Because basically what basically they usually show you like organism. The actual size is what the size of the organism actually is. The image size is what it is in the picture, and the magnification is how much the image has the actual thing has been magnified in the image. Because sometimes it looks smaller or bigger. Usually in most cases it's bigger, but sometimes it's even smaller. So you actually have to see, and this is very important. The conversion scale is very important because in IB you're always strict with the conversion. So make sure that you know the conversion very, very well, and make sure that you know this, because then you remember all the formulas. Because for example, let's say you want the image size, you just cover the image with your finger, and then you see A times M. If you want magnification, just cover your finger with M, and then you see I over A. This is the best trick for it, and you're actually able to do this. But again, please remember to do practice. These needs practice because there's many tricks when it comes to this, and you also have to know your units. Now for the emergent properties. So basically, emergent properties arise when the interaction of individual components produce new functions. Basically, you know, some, like in Aristotle's words, so, you know, it's a one thing, like the thing is a greater sum of its parts, basically. So multicellular organisms are able to complete functions individual cells or unicellular organisms are not able to undertake, because of you know, their different components. So basically, cells are grouped to form tissues because tissues are a group of cells. Then organs are formed because organs are a group of tissues. So then organs are then formed from the functional grouping of these tissues. And then organs may interact and form organ systems. Organ systems are made out of what? Organs, capable of carrying out specific body functions. And then organ systems collectively carry out life functions of the complete organism. So as you can see, this is it. Cells are a group of tissues. Tissues are a group, tissues are a group of cells. Organs are a group of tissues, systems are a group of organs, and the organism is a group of systems. You have different systems, and this image is from BioNinja. So now let's discuss cell differentiation. So basically, what is differentiation? Differentiation is the process during development whereby newly formed cells become specialized and distinct from one another as they mature. So I'm going to actually explain this deeply when I actually read the next point. So basically, all diploid cells of an individual organism share an identical genome. So each cell contains the entire set of genetic instructions for that organism. But the most important part of this, really important, is that not all genes are expressed in all cells. Meaning that, for example, in blood cells, that specific gene is expressed for it to be a blood cell. It does have all the other genes, but just that one gene is expressed. And the fewer the genes, or the amount of genes that are expressed, the more specific and differentiated that cell type will be. And every single time they ask you in paper 1 questions, what makes the cell like specialized, you have to say that they have a specific gene that's expressed, because they do contain all the genes, but specific ones are expressed. Always remember that. And the activation of different instructions, which are actually genes within a given cell, by chemical signals will cause it to differentiate. Now, let us discuss gene packing. So basically, within the nucleus of a eukaryotic cell, DNA is packaged to form chromatin. So in newly formed cells, they receive signals which deactivate more rarely activate genes. So basically, active genes, they have euchromatin, meaning that the fewer active genes a cell possesses, the more specialized it becomes, like I previously mentioned, and resulting in a cell's metabolism and shape to change because it actually carries out specific functions, because not all cells look the same. And inactive genes, they're named heterochromatin, and they have a more condensed form. So differentiated cells will have different regions of DNA packed as euchromatin and heterochromatin according to their specific functions. So basically, like I'm going to use the blood cell again. So basically, it's going to have euchromatin for the part that's activated, for like the blood part, and then for the rest, it will have heterochromatin. Now we're going to discuss specialized cell. So all specialized cells and the organs constructed from them have developed as a result of differentiation. So basically, organs, they're a group of tissues made out a group of tissues which actually perform a particular functions, but tissues, they're a group of cells that actually perform a specific function. So now unspecialized cells, basically they can continuously divide and replicate, and they have the capacity to divide into special cell types. This is the most important part. Key qualities of unspecialized stem cells. So firstly, self-renewal. So basically they have the ability to continuously divide and replicate. And second, potency. They have the capacity to differentiate into specialized cell types. This is very, very important because it's very, very highly asked about in the IB. So firstly, in totipotent, what it means that they can differentiate into any cell type. So for example, a zygote. Pluripotent is that they can differentiate into many cell types, not all, but many. So for example, it's an embryo. Multipotent is that they can differentiate into a number of closely related cell types. So it's kind of limited. For example, a blood stem cell. And unipotent is that they cannot differentiate, but are capable of cell renewal. An example is a muscle cell stem cell. And this is actually an image from biology, which actually helps you understand. Now, the cost of cell specialization. So specialized cells are efficient at carrying out their particular function, such as transfer, support, or protection. And by specialization, increased efficiency is received, yet the specialized cells are now dependent on the activities of other cells. So there's actually, now we're going to be talking about acellular organisms which do not conform to the cell theory. So acellular organisms are multinucleated organs or organisms, and they are not divided into separate cells. For example, we have striated muscle fibers. They're multinucleated. They have more than one nucleus, but they're still unicellular, and they do not conform to the cell theory. And this is actually very asked about in the IB, especially in the paper 1 questions. They're like, which one is the exception to the cell theory? So always choose the choice that is striated muscle fibers. So now, stem cells are necessary for embryonic development. So this is how it goes. So first they're a young cell, then cell division takes place, and then cell growth takes place. Then there's cell specialization, and at that point, what happens is that the cell begins to mature or it's already matured, and it cannot divide, so then it ends up dying. Now, what is a stem cell? Now, what's the definition? Again, stem cells are the most important part of this whole chapter. You have to focus on them especially. So basically, a stem cell are cells that have the capacity for repeated cell division and remain undifferentiated, and subsequently have the capacity to develop into mature cell types. Now, stem cells are the actual building block of life because they divide and form cells that divide into a range of mature cells of organisms. And stem cells, they're found in all multi-cellular organisms. Basically, many tissues in our body, they have stem cells. At the next stage of embryological development, cells lose the ability to divide as they develop into tissues and organs of the organism. And embryonic stem cells, they are capable of further division. Now, what are embryonic stem cells? They're undifferentiated cells that have continuous division and can develop into all types of cells of an adult organism, any type. They make up the bulk of embryo as it commences development. Now, adult stem cells have undifferentiated cells that are capable of division. But that's very important, they give rise to a limited range of tissues. This is the difference between adult stem cells and embryonic stem cells, is that embryonic stem cells can differentiate into any type, whereas adult stem cells can only differentiate to a limited amount. And adult stem cells occur in the growing adult body. They replace damaged or dead cells within the organ. So now, what is stem cell therapy? So stem cells can be used to replace damaged or dead cells with healthy and functioning ones. So what does this process actually require? So firstly, the use of biochemical solutions. What they do is that they trigger the differentiation of stem cells into the desired type. So basically there are like these like slashes between because they're from two different resources. So I'm just going to use the first resource, which is from BioNinja, because it explains things much easier. Then the second point is that there's surgical implantation of cells into our own patient's body. But what happened is that suppression of the host immune system has to take place because we have to actually prevent the rejection of these cells because these cells might be from a foreign source, such as they might be, from another patient's body, from an adult, it might be an adult stem cell. And there must be careful monitoring of the new cell to ensure that they do not become cancerous. Okay, so now medical conditions identified with stem cells technology, which may have potential to actually bring relief or cure. So firstly, Stargardt's disease. Basically, what happens is that there's a breakdown of the light-sensitive cells in the retina. It does not actually ruin or destroy peripheral vision, but it actually causes blindness, so the patient won't actually be able to see. And it is an inherited condition due to a mutation in the gene with the processing of the vitamin A in the eye. This is very important, it's very asked about. So what's the treatment? The treatment is that the embryonic stem cells are being used to regenerate damaged light-sensitive cells. So what happens that when these light-sensitive cells are broken down, stem cells actually are used to differentiate into them, and then these patients get these cells back. And Parkinson's disease. So basically, Parkinson's arises from the death of neurons, which are nerve cells of the brain that control subconscious muscle activities by the means of the neurotransmitter called dopamine. Now, what is dopamine? Dopamine is a neurotransmitter that's responsible for transmitting signals involved in the production of smooth, purposeful movements. So basically, what ends up happening is that they have these movement disorders, such as limb rigidity, and they don't have a balance. They're not being to walk in a balanced way. They have impaired balance because their neurons are dying. They're not having control of their movements anymore. Then they have cardiac muscle damage. So basically, it's death of muscle fibers due to myocardial infarction is caused by major interruption of blood supply to the area of the cardiac muscle. So basically, this does not have a treatment, or at least there's the resources do not provide any treatment for this. At least I did not find anything that actually provides and that stem cells actually use to provide treatment for this. But for type 1 diabetes, it arises when the B cells of the pancreas are destroyed by the body's immune system because there's a severe lack of insulin results. So basically, insulin, what's insulin? Insulin is a hormone which naturally maintains the blood's glucose concentration at about 900 milligrams per centimeter cubed. So what happens is that there's a severe lack of insulin in diabetes. And in diabetics, the number of glucose is not controlled and becomes permanently raised, and it's kind of irreversible. So what happens is that using stem cells, there's the replacing non-functioning islet cells with those capable of producing insulin types in type 1 diabetes. So basically, what they do is that they actually end up replacing all of the islet cells with ones that can actually produce insulin, and they're gonna actually start having normal insulin concentrations in their body. So now leukemia. So basically, it's cancer of the blood or bone marrow, resulting in abnormally high levels of poor functioning blood cells. So what happens is that hematopoietic stem cells are harvested from the umbilical cord, peripheral blood, or the bone marrow. And then the second thing that takes place, like the second step, is chemotherapy and radiotherapy. They're used to actually destroy the white blood cells that are cancerous. Third of all, the hematopoietic stem cells, which are, but they're actually abbreviation. The abbreviation is HSC, are transplanted back into the bone marrow, and the HSCs are differentiated to form new healthy white blood cells. And again, for burn victims, they graft new skin cells to replace the damaged tissue. And this image is from BioNinja. So the sources of stem cells, basically, there's embryo, and the embryo can be actually created by therapeutic cloning. There is a certain adult tissue like bone marrow, but the cells are not pluripotent. And there's umbilical cord or placenta of the newborn baby. And now these are some comparison of stem cells that you can actually go over. They're from Bionology, and they're on my Google Classroom. So if the link's down below, if you'd like to see my Google Classroom, it has certain notes and so. And then we have to discuss the arguments of therapeutic cloning. So basically, stem cell research may prove, so basically, arguments for and arguments against. So the arguments for, basically, stem cell research may pave way for future discoveries and beneficial technologies. So it provides health benefits, and these like certain discoveries would not have taken place or been discovered if the therapeutic cloning had been banned. It may be used to cure serious diseases or disabilities with cell therapy, like I talked about, Stargardt's, leukemia, skin burns, Parkinson's, all of these. So again, more health benefits. So what happens is that they replace the bad cells, like for example, in diabetes 1, the islet cells, with new and functioning ones. And then transplants are less likely to be rejected as they are cells which are genetically identical to the parent. So again, because these cells, they can differentiate into any type of cells. And transplants do not require death of another human. So again, it's ethical. Stem cells can be taken away from embryos that have stopped developing and would have died anyways. So both, such as abortion. So basically, both ways, these stem cells were going to die. So why not use them for something good instead of letting them die, because they're just going to waste. Stem cells can be taken away from the embryos that have stopped developing. Oh yeah, I just said that. Cells are taken at a stage when the embryos have no nervous system and can arguably feel pain. So basically, these embryos, they don't have any nervous system, so they actually don't feel any pain. So it's useless to actually not use them because they don't feel pain. So it's completely ethical. And stem cells can be created without the need for fertilization and destruction of natural human embryos. So they don't have to actually create human embryos. So again, it's not unethical. So arguments against therapeutic cloning. So firstly, it involves the creation and destruction of human embryos, which is wrong against ethics. Embryonic stem cells are capable of continued division and may develop into cancerous cells and cause tumors. More embryos are generally produced than needed, so excess embryos are killed, so some are actually going to waste that way. With additional cost and effort, alternative technologies may fulfill similar roles, such as nuclear programming. So basically, they're saying that you're using stem cells, which can actually, turn into something living, but instead you can actually use other technologies which don't have any ethical issues, and they can fulfill the same purposes. And then religious or more like objectives, because you're in some religions, it's considered killing a life because it's an embryo and it has the potential of life, so it's considered killing a life. And the embryo which is created would potentially be used in IVF and develop into human fetus. So again, it can actually end up being a human fetus. So this is it. I don't think we have time for the paper 2 questions. Maybe another video. If you guys want a video for paper 2 questions, please tell me. You guys can join my Google Classroom, which actually has these notes and other resources and other notes and all the resources I actually used for this video. That way you can actually delve deeper into them. And that's it. If you have any questions, comment down below and you can ask me on my Google Classroom. Thank you so much for watching. Bye-bye. Enjoy your day, guys. And really good luck, especially for the ones that have the IB exams. Good luck. Bye-bye, guys.